how's your day? I'm sure that we had a lot of fun with our previous lesson. I know that we are very excited to learn more knowledge today. By the way, I am Teacher Sharon Lombarda. I am your teacher in General Biology 1, and you may call me Teacher Sheng. I am also pleased to bring you another learning adventure. So today, we are going to discuss mitosis and meiosis. Here are our objectives for today's lesson. Describe the stages of mitosis and meiosis. Compare mitosis and meiosis and their role in cell division. And explain the significance or applications of mitosis and meiosis. Before we proceed to our lesson, let me ask you these questions. Did you already experience having wounds? What happened to your wounds after a few days? Why do you think you don't have the same looks with your own parents and siblings? Let's find out how this happens in this episode. To begin with, let us recall the different stages in the cell cycle.
The cell cycle is the complex sequence of events by which cells grow and divide. In eukaryotic cells, this process includes a series of 4 distinct phases. These phases consist of the mitosis phase, gap 1 phase, synthesis phase, and gap 2 phase, collectively called the inner phase. During the gap 1 phase, the cell increases in mass and organelle number in preparation for cell division. Animal cells in this phase are diploid, meaning they have 2 sets of chromosomes. In the synthesis phase, the chromosome content is doubled. For the gap 2 phase, the cells synthesize additional proteins and continues to increase in size. After the inner phase, the cell is ready to enter in the mitosis, which has 4 stages or phases. These are the prophase, metaphase, anaphase, and telophase.
We have just reviewed the stages in the cell cycle and the important events in each stage. We are now ready to discuss our lesson for today, which is all about the 2 types of cell division, mitosis and meiosis. At this point, I would like you to watch a video clip presentation. In watching the video, please take note of the important concepts being presented on the differences and similarities of the 2 types of cell division.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis. But two events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
There you have it! you have just watched the video presentation that highlights the differences and similarities between the two types of cell division. I believe that you are now ready to answer these questions. How many nuclear divisions does mitosis have? How about in meiosis? You are right. Mitosis has one nuclear division while meiosis has two. Do you think there is a synapse or crossover in both cell divisions? Exactly! There is a crossover only in meiosis but not in mitosis. In what organisms do mitosis and meiosis occur? Great! All organisms undergo mitosis except viruses, while in meiosis, it occurs only in animals, plants, and fungi. What type of cells are produced in mitosis and meiosis? Definitely, mitosis produces body cells, while meiosis produces germ cells. Describe the number of chromosomes under mitosis and meiosis. Yes, the number of chromosomes stays the same in mitosis, while in meiosis, only half the number of chromosomes from parent cell develops. How many daughter cells will be produced in mitosis and meiosis? Correct. Mitosis produces 2 daughter cells, while meiosis produces 4 daughter cells. How do the daughter cells produced in mitosis and meiosis differ? Very good. In mitosis, daughter cells are genetically identical, while in meiosis, the daughter cells are genetically different. What type of reproduction happens in mitosis and meiosis? Perfect! Asexual reproduction happens in mitosis, while sexual reproduction takes place in meiosis. If a mother cell has 6 chromosomes in mitosis, how many chromosomes are there in a daughter cell? Correct! There are 6 chromosomes in each daughter cell after mitosis. How about in meiosis? How many chromosomes are there in a daughter cell if the mother cell has 46 chromosomes? Excellent! There are 23 chromosomes in each daughter cell. Remember, class, that in mitosis, there are 2 daughter cells being produced, while in meiosis, there are 4 daughter cells. Did you know that in 2018, the University of Zurich has discovered a new control of cell division? It stipulates that when a cell divides, its constituents are usually evenly distributed among daughter cells. Researchers have now identified an enzyme that guarantees that cell constituents which are concentrated in organelles without membrane are properly distributed. Their discovery opens up new opportunities for the treatment of cancer, neurodegenerative diseases, aging process, and viral infections. Congratulations, dear learners! You did a great job in today's lesson. A big thumbs up to everyone. You now have a clearer understanding of the 2 types of cell division. Now, you have realized that wounds heal through the process of cell regeneration, which replaces dead and destroyed cells. This happens during mitosis. On the other hand, we don't look exactly the same with our own parents and siblings because of the chromosomes crossover during the meiosis, resulting to diversity. we have successfully discussed our lesson for today. Before we end, let us have the salient points to remember about cell division. Mitosis is a form of eukaryotic cell division that produces two daughter cells with the same genetic component as the parent cell. Chromosomes replicated during the S phase are divided in such a way as to ensure that each daughter cell receives a copy of every chromosome.
In actively dividing animal cells, the whole process takes about 1 hour, while meiosis is the form of eukaryotic cell division that produces haploid sex cells or gametes from diploid cells. The process takes the form of one DNA replication followed by two successive nuclear and cellular divisions, meiosis 1 and meiosis 2. As in mitosis, meiosis is preceded by a process of DNA replication that converts each chromosome into two sister chromatids. Therefore, mitosis and meiosis are responsible in cell development and in determining the different traits of human and other living things. Now, I would like you to see yourself in a mirror. List down your observable traits, after which recall or look at your mother and father. Which of their traits do you think have you inherited? What particular traits are you different from your brothers and sisters? Do you appreciate these traits that you have in common with your parents and siblings? What should you do with the traits you find exactly different from them? Another fruitful discussion with you has just ended. If in case you still have queries regarding the lesson, feel free to contact or call your subject teacher for clarification. That would be all for today. See you all in our next learning escapade.